Thank you, Dr. Shankar, for that wonderful words. I would like to thank the organizing secretaries and the members for inviting me to share my scientific data with you all. I work for a School of Regenerative Medicine. It is one of the kind of institute which dedicated for stem cell research, and it's located in Bangalore. It comes under Mahi Manipal Academy of Higher Education. Here I'll be sharing some of the scientific preclinical data, mainly focusing on the temporal lobe epilepsy model. Can I have my slides, please? So the temporal lobe epilepsy, it's a kind of a common epilepsy. It affects more than 65 million people all over the world, and in India alone we have somewhere around 6.5 million people affected with this disease. There are different types of epilepsy, but temporal lobe epilepsy is a kind of common type. And there is always a sequel can be observed in the temporal lobe epilepsy condition. That is an initial precipitating injury. This may be status epilepticus, status epilepticus, and stroke, trauma, or kind of high fever. Following this initial precipitating injury, there is a kind of silent period it varies in time. For example, in animal model, we observe that it is around 2 months. And following that, there is something called a spontaneous recurrent motor seizure. This, once it's this SRMS originates, till the animal die, we can see this. It is similar to human, what you see, the temporal lobe epilepsy patient. So this period, the silent period, we call it as epileptogenesis. And it is not a silent period actually. There are so many intracellular changes happens following this initial injury. And that, for example, we can study with the use of animal model. We cannot get a sample from a human subject. So we have a wonderful prototype for temporal lobe epilepsy using electrical stimulation, or you give a chemoconvolution drug. And what we adapt here is that we use a neurotoxin called kynic acid at a higher dose. When you inject to this animal, they develop status epilepticus. This is the initial precipitating injury. And they will undergo a latent period. Can I have that video played? Yeah, this is how it will, when you inject this kynic acid to this mouse, they will have the epilepsy, the seizure. And following that, they enter into the latent period for approximately 2 months. And they develop this spontaneous recurrent motor seizure without any drug stimulation. So this is a wonderful model. Once they have the status epilepticus, when you sacrifice them, cut the brain and look into the various parts of the brain region, especially in hippocampus, you can see an enormous amount of neuroinflammation. What you see here on the top panel, the green color one, is an indication of how severe the inflammation following status epilepticus as compared to the control animals. It's not only the neuroinflammations, they also started showing signs of neurodegeneration as early as 72 hours following the status epilepticus. You can see that in the morning, Dr. BSSR Rao talked about the hippocampus and its function and its structural organization. In the status epilepticus, following the status epilepticus, there is a significant neuronal loss. There is a significant neuronal loss in the CA3 region and the dentate hilus. If you allow the animal untreated, the neurodegeneration spreads to other part of the hippocampus. As you can see here, the neuronal loss completely gone here in this area. So, it's also important to mention here that now it is well standardized that even at the adult age or even at the very older age, there are new neurons formed. The dogma that, once you reach certain age, no neurons are formed is gone. There are certain areas in the brain region which can produce new neurons throughout your life. These neurogenic regions, one is in hippocampus. And here, what you see here, this is the newly born neurons in the hippocampus. Following status epilepticus, you can clearly identify that this, in the normal condition, it is properly oriented in the molecular layer, whereas following status epilepticus, they slips down. And this is wrong, actually. So this what will happen is it will lead to an inappropriate neuronal connections which can lead to subsequent seizure attack. Another thing is that in the CA3 region, whatever the neurons that are there in the granule cell layer, the MOSIF fibers that connect to the CA3 region, they generally terminate here. But after status epilepticus, as you can see here, they terminate on their granule cell neuron itself. So it is like having a recurrent loop on their own head. Whenever they get excited, they keep on giving excitation that leads to the seizure behavior. And over time, what will happen is they lose the interneurons. Interneurons are very important for your neuronal survival. It balances the excitatory activity. So following the status epilepticus, as you can see here, that there is a complete, not getting oriented with. So you see the neurons, these are all interneurons in the hippocampus. Following status epilepticus, they are completely wiped off. So we have a model here that is wonderful. It replicates most of the phenotype of human temporal lobe epilepsy. So what is the treatment possibilities available? Of course, there are anti-epileptic drugs available for treating temporal lobe epilepsy. But 30% of these patients are refractory to anti-epileptic drugs. They do not respond. Even the people who are responding to this anti-epileptic drug, they only have a symptomatic relief. They have significant other side effects. When the epileptic activity is uncontrollable, doctors suggest for a complete removal of the seizure foci, which by itself have a lot of other side effects like cognitive impairment, motor impairment, and other things. That's where the stem cell comes into picture. I will not go into detail about what is stem cells. You have been heard enough about stem cells. So here I'm going to talk about type of stem cells. This is the embryonic stem cells, which can be derived at different stages of development. It can also be derived from adult, that from morning we have been hearing. So the disadvantage of embryonic stem cell is that there is a lot of ethical constraint. And another potential thing is these stem cells, embryonic stem cells, are very potential, but at the same time they are very notorious. If you don't know how to control their proliferation, they end up in forming teratoma. The teratoma you might have heard about, that it will have every cell possibility. It will have liver cells, kidney cells, hair cells, dentin, everything will be there in a single mass. The other thing is that the immune rejection, the graft rejections. So in search of the possibilities for other stem cells, the people come up with the adult stem cells. We have heard about adult stem cells. There are different types of adult stem cells, bone marrow, dental pulp, you can derive from umbilical cords. So these adult stem cells, it can be used as an autologous or allogenic stem cells. And this, since yesterday night, when I searched in the clinical, there is a website called clinicaltrial.gov. There are 896 clinical trials currently going on using these adult stem cells. And they are at the different phases of the clinical trial. So among these stem cells, adult stem cells, bone marrow stem cells was the gold standard stem cells. It has been used for every diseases, whether it is a neuronal or non-neuronal, it has been used, and there is some amount of recovery. But there is a question that in the morning when the discussion came, identifying an appropriate stem cells for a kind of different disease condition is the most challenging. So there is a need to find some stem cells that comes from the ectodermal origin where the neurons and the glial cells are produced. Another disadvantage of this bone marrow mesenchymal stem cell is that it's a highly painful process. Finding a donor is very difficult. You have to use anesthesia. There are some possibility that even the donor may die. So in search of that, for a neural based thing came the dental pulp stem cells, which comes from the ectodermal origin, and it's a neural crust cells. It migrates all over the body. It's there in the skin cells. It is also reside inside the dental pulp. So we decided to use the dental pulps to study whether it can protect the early hippocampal changes happening following the status epilepticus. Our long-term goal is, once we identify that it is protecting, we want to see whether it can reduce the seizure intensity, it can increase the cognitive impairment and the other comorbidities relevant to temporal lobe epilepsy like anxiety, depression. So we are at the very initial stage. So these stem cells, the dental pulp stem cells, if you see, they have similar to bone marrow stem cells. They adhere to the plastic, the culture plastic thing. They express some of the MSC markers. They do not express any hematopoietic markers. And you put it under a proper condition, culture condition, they can differentiate into adipocytes, chondrocytes, and osteocytes. What is the dissimilarity between the DPSC, the bone marrow and DPSC is that these DPSC cells express some of these neural curse markers, whereas the bone marrow only few, 1 or 2 SOX9 and twist can be found there. And another thing, even in a normal MSC culture condition, you just grow this BMMSC and DPSC and put a staining for something relevant to neuron, you can see that the beta-3 tubulin, which is generally used to identify a mature neuron, they express more in number compared to the bone marrow stem cells. And when we did a kind of growth factor profiling, taking the mRNA and doing the real-time PCR, we can find that there is a significant fold increase in the growth factor expression in the DPSC as compared to bone marrow cells. So given this, we want to study, is there any priority between the DPSC and BMMSC in migrating towards degenerative conditions? So we did an experiment in which the lower bottom of the culture plate was cultured with hippocampal neurons. They were treated with neurotoxin, the same neurotoxin that can induce status epilepticus. And on the top, we put a culture insert which will have a micropose. And these cells, if they have a kind of special attraction towards the central nervous system, they will migrate more. As you can see here, when we did the transfer migration assay, in a degenerative condition, the DPSC migrated much more than a BMMSC cells. And here is the quantification data. We also studied whether they can migrate through the extracellular matrix, which is also important. For example, when you are injecting these cells through intravenous injection or any systemic injection, there is a problem that they get entrapped in the lungs. The blood vessels are very tiny that they get entrapped, and most of them die, some migrate through. So we want to know whether this DPSC has a better migration towards the CNS thing. So in a kind of in vitro condition, to avoid the number of animal sacrifice, we studied in this condition. And we did the matrigel. Matrigel is something like that mimics the extracellular matrix. So there also we found that the DPSC have a better migration than the BMMSC. We did a membrane migration because they have to pass through the blood-brain barrier when we are giving systemic injection. So what we did experimental settings where we can collect this chorion allanto membrane from the egg and just place these cells, the DPSC or BMMSC, and see whether they can migrate through this membrane. And here also we can see that the DPSC has a better migration. So we have a proof of principle that the DPSC have a better migration, they express more neuronal related growth factors, they express neuronal markers. Now we want to study whether in a kind of in vitro neurodegenerative conditions, whether they can protect the neurons. So for that we used the hippocampal cells which express most of the glutamate receptor profiling. And when we apply this, the hippocampal cells with kinic acid, they die depending upon the dose. And these hippocampal cells, they express all these neuronal markers, they do not express the glial markers. So once we establish this, we want to study this in vitro assessment. So before going to that, I want to explain that there are 3 major hypotheses that is prevailing in the mechanism by which the stem cell therapy could, the functional output could happen. One is the transdifferentiation. You transplant a cell either in a kind of in a brain or in a liver area, and they transdifferentiate into the local phenotypic cells. The other possibility is that they may be simply sitting there and producing some of the growth factors and cytokines that can stimulate your endogenous residential stem cells. The third is that these healthy and energetic stem cells can donate their organelles like mitochondria, Golgi apparatus, or endoplasmic reticulum to dying cells and thereby protecting them from further degeneration. So these are the possible hypotheses that is existing now. And we were interested to see whether a direct cell-to-cell contact or a secretome alone is, the growth factor alone is sufficient to do this neuroprotection. So for this, we set up an experimental condition wherein we growed these hippocampal cells and they were treated with the neurotoxin kinase acid. And what we did is we co-cultured them with DPSC or BMMSC, or what we did is we just took their culture condition, the medium. Generally, these mediums are thrown in the trash as a normal cell culture process. But what we found is that these cells inside the body or in a kind of tissue culture condition, they constantly exchange proteins in the surroundings. So they should, this culture condition, whatever we are discarding, should have a valuable proteins that can protect the neurons from the kinase acid toxicity. So what we did is we collected this condition medium, we put that culture condition medium in this neurodegenerative condition, and we did a cell viability assay. As you can see here, this is the kinase acid treated one, this is the co-culture treated, and this is the condition medium. They have equal neuroprotection. So that says that there is no need for transplanting cells for neuroprotections. You can utilize these secretomes to have a neuroprotective effect. This is something called stem cell based therapy. So what we did is now we have in vitro evidence, but whether it will work in an in vivo condition, that we want to study. So next we move on, and what we did is we lesioned the hippocampus using the same toxin, kinase acid. And what we did is, after some time, we transplanted this DPSC or BMMC directly into the lesion area. In another set of animals, we collected the condition medium, we infused that condition medium into the hippocampus. And we allowed the animal to live for 30 days, and then we did the cognitive assessment because it is the hippocampus that is primarily affected in the temporal lobe. There are other regions, but particularly hippocampus is more affected. And you can see whether your therapy is working or not by doing some cognitive assessment or hippocampal dependent tasks to see whether your therapeutic intervention is working. So what we did is we did the radial maze assessment, wherein this animal will be semi-starved and they will have a reward, a choco pop or something like that, they will have a reward. The task is that they have to go into this arm, have that reward, and they should never re-enter this arm. That's some kind of memory assessment. As you can see here, the red dots, that is the kinase acid treated one, they never improved that in the task. Whereas here, the bone marrow MSC and DPSC or their respective condition medium showed significant improvement in the memory performance. So after they reached a criteria of 95%, we let them live in their respective home cage. After 30 days, we brought them back to the experiment condition setup, and we just want to measure whether they still remember that, and they do remember that. So given that, we sacrificed the animal, we looked into whether there is any neuroprotections. As you can see here, in the kinase acid treated one, the CA3 neurons are lost, whereas in the BMMSC, DPSC condition medium treated, there you can see the intact CA3 regions. It's not only the neuroprotection, the inflammation came down drastically as compared to the lesioned one. But what we observed is that there is a difference in the condition medium derived from BMMSC and DPSC, that the BMMSC condition medium did not reduce the inflammation much as compared to the cell transplanted one, whereas the DPSC and the DPSC condition medium had a similar effect. Next, we want to see whether this condition, this stem cell transplantation, can activate the residential stem cells. As you can clearly see here, you can see here that the BMMSC condition medium injected here, this is the normal one, this is the kinase acid treated one, this is the stem cell transplanted or condition medium treated one. You can see a huge increase in the neurogenesis. This is very important actually. It helps in cognitive improvement, it helps in reducing the depression, which is normally found in temporal lobe epilepsy patient. So as you can see here, there is a significant increase in the neurogenesis. And between this BMMSC and the DPSC, if you see, the neurons, the newly born neurons that are formed in these areas, they are very well developed. They have more branches. So that says that the culture medium, what you are giving, help the endogenous stem cells to proliferate as well as mature and reach their destiny. So this is a kind of very significant thing. And once we identified that the culture medium have a similar effect like cell transplantation, now we want to know that whether you need the total conditioned medium or is there any small fraction in the conditioned medium that is doing this neuroprotective activity. So further what we did is we took the culture medium from the BMMSC and DPSC, and what we did is we filtered them based on their molecular weight. And whenever we filtered and whatever the filtered product we have, we assessed it in an in vitro neurodegenerative condition to see whether they have that neuroprotective ability. So as you can see here, the product which is above 100 kilodalton, they do not have this neuroprotective activity. In fact, even a normal neuron they were killing. Then we went further to filter them based on the molecular cutoff filter between less than 100 kilodalton to 10 kilodalton. And here also we did not find any neuroprotection. We went further and filtered. It falls in the range of 10 to 3 kilodalton where we could able to see there is a neuroprotection. So you don't need a conditioned medium, total conditioned medium. There are small molecules which falls in the range of 10 to 3 kilodaltons which can able to recapture your entire stem cell therapeutic benefits. So these small molecules, nowadays a lot of studies coming out that the stem cells also release some of the vesicles as a normal mean of communicating with other cells. This is not only specific to stem cells, any cells in the body, they release these extracellular membrane vesicles that can carry protein, mRNA, siRNA, and RNA which can go and bind to the host genetic material and it can bring about significant functional improvement. So these exosomes are something like a nano-sized structure which can be isolated by ultracentrifugation. You have to collect the conditioned medium, subject them to ultracentrifugation, and you can identify, you can get this one, and there are markers available to identify these exosomes. So some of the markers we have done it here, they correspond to these exosomes.
Dr. Anandh, you have 2 minutes. Will you please wind up?
2 minutes? Okay. Can I have the... So this is... No audio? So this is the exosomes that comes out of the cells. Not only in stem cells, in any cells, these exosomes can carry these valuable genetic materials. So your stem cells after transplantation, they are basically not trans-differentiating into neurons or astrocytes, but they are releasing growth factors and these kind of exosomes which can bind to your host cells and activate their reparative mechanisms. They can activate their gene response for growth factors and thereby they can bring about the neuroprotections. So we extracted these exosomes, we labeled them with a dye, whatever you see there is a red color, those are the exosomes, and we want to see how they are localized in these cells. So in an in vitro hippocampal culture, we just have applied these exosomes and we could able to see that there is an internalization of these exosomes. These exosomes are also present in the membrane vesicles. Given this, we did a cell viability assay. As you can see here, the exosomes derived from the DPSC and BMMSC have a similar cell survival ability, neuroprotective ability. And they also induce the growth factor PDNF, which is essential for this hippocampal neuronal survival. They activate the cell survival pathway like PI3K, and they increase the expression of anti-apoptotic factors like BCL2. Okay? So once we found out that again these exosomes, they are very potent, we have to restrict their extraction. We have a cells, for example, we have a vial of DPSC. Can I exploit them as much as possible to extract these exosomes? No, there is a restriction. You can go up to passage 3 to 4. Beyond that, if you start collecting these exosomes, even in a normal neuronal culture, they start killing these cells. And these exosomes, they lose their neuroprotective ability if you increase the dosage. There is a dosage range also. So once we found out that what we did is, these exosomes are nano-sized, what we did in the same animal model, we allowed them to get into the brain through your nasal route. This is the easiest route, and we could successfully able to get them into the various brain part. As you can see the red dots, these are the exosomes that were injected through the nasal routes. So then we did a kind of histological analysis for the neuroprotections. One important thing we found is that these exosomes, unlike the cell transplantation or condition medium treatment, they only provided a partial neuroprotection. So we could not able, whatever the red line you see, there is no recovery, whereas the green line, then cells are protected here. So what it says is that we still have to work on the dosage and the time, how long we have to treat them, all those things. This is a very pilot study that we are currently doing. And this is the conclusion that, this is the topic of my talk, that from billions to narrow. That's why you don't have to transplant billions of cells to do the neuroprotections. You can use these nanocells exosomes, which may be a future stem cell based therapeutic approach. And there is still a caution that it's at a very initial stage. We don't know whether they carry some of these oncogenes, which like stem cells, embryonic stem cells, can also induce cancer or teratoma that needs to be identified. And coming to the thing, this is my group which work in my lab. And we in SWORM, the School of Regenerative Medicine, we have masters in regenerative medicine. We have MPhil and PhD with fellowship facilities available. Some of them are the master degree students and PhD students. I would like to thank our institute, Manipal Academy of Higher Education, for providing this platform, and also the funding agency. Most of our study is funded by DPT, ICMR, and CSAR. Thank you very much.